So, biocatalysts can replace chemocatalysts in synthetic routes. Okay, this is the easy thing to do, I think. So what chemists increasingly do in academia, industry, the generic companies are very keen on this. They look at synthetic routes. So this is the API for the generic drug Singulair, and it's a relatively complex molecule, but it's got this chiral asymmetric center here. One of the key precursors is an alcohol. Classically, that alcohol was generated by asymmetric reduction of a ketone using stoichiometric chiral reducing agents. And for the past few years, I would say, there's been a lot of interest in asking the question, can we replace a sort of rather unpleasant chemical reagent with a biocatalytic step? Okay, now that's easy in the sense you just replace one reagent with another biocatalyst. And so the way this is done now is it's possible to find enzymes that do this, and it's possible to do cofactor recycling. So the whole technology has moved on massively to the point where a lot of people in the pharmaceutical industry tell me now if they want to do a ketone reduction, they start off with an enzyme and then they perhaps move to a non-biocatalytic approach. So the whole approach has completely changed from chemo to biocatalysis. So that's very interesting, and that would be nice if in the next few years that became true of a broader range of technologies. And then you can read very nice papers, particularly by Codexis, who publish data where they go through all of the parameters for the biocatalytic and the stoichiometric process, and they argue why the biocatalytic process is better, and in terms of not just catalysis but the temperature, the use of solvents, which we've heard a lot about before lunch. And this is my impression of biocatalysis. Often it ticks several boxes. So it might be cheaper starting material, more environmentally acceptable, better uses of solvents. But what I would argue is more exciting, certainly from my perspective, is that you use biocatalysts to make and design new synthetic pathways. You don't just do replacement chemistry. You think about making a product in a completely different way because of the availability of biocatalysts. And that could, if it's shorter, more efficient, more sustainable, that could offer even more benefits. So here's a nice example from DSM. So this is an API called perindopril, antihypertensive. A key building block is this molecule here with 3 asymmetric centers. The classical approach to that is a Fischer-Indol synthesis. So you essentially use the Fischer-Indol synthesis to build up a precursor which you then make in racemic form, and then you do a sort of a classical resolution. And obviously you lose half the material en route. So the DSM route redesigns that approach. It takes advantage of a asymmetric amination using an ammonia lyase to put in this amino group at the alpha position, which is interesting because that's sort of intuitive in terms of the reactivity of the cinnamic acid. And then you do a copper catalyzed intramolecular amination reduction. So you've got a completely new way of making that molecule that sort of is so much more efficient than the classical Fischer-Indol synthesis. So for me, particularly as an academic, this is the future, is making molecules more efficiently, but it relies on having more enzymes to do more synthetic steps than we currently have. So theme 3, which DSM touched on, is actually we're not just going to use bio or chemocatalysis. Probably we're going to use combinations of bio and chemocatalysis, maybe one step after another, maybe even together in the same reaction. And people are obviously developing ways of doing this, and that's very exciting because you use the benefits of bio and chemocatalysis. So I think that's going to be, it is a major theme that will increase. I'll give you an example of this. This is from our work. These are very important drugs that were launched about 18 months ago by Johnson & Johnson and also Merck. They are new treatments for hepatitis C, telaprevir, which I understand is the fastest selling drug ever. The sales of telaprevir, I think, were $1 billion in the first, or €1 billion in the first 12 months, because essentially these drugs cure large numbers of people of hepatitis C. So we were interested in making the central bit, but particularly with this idea of combining chemo and biocatalysis. So we first of all worked on the biocatalysis. We engineered an enzyme called monoamine oxidase to do a desymmetrization of this fairly cheap symmetrical starter material to make this chiral imine, which interestingly trimerizes, and you can recrystallize it to optical purity. So that was the biocatalysis bit. And in fact, you can take that imine and you can make those that building block via a sort of fairly classical synthesis by just elaboration of the chiral imine. But we wanted to do something a little bit more interesting to sort of build in the chemo synthesis part, and so this was a very interesting collaboration with the Free University of Amsterdam, who are part of Chem21, where we took the biocatalytic building block and developed a highly convergent synthesis using their expertise in multi-component chemistry. So one of the reasons we were keen to get the Free University of Amsterdam into Chem21 is they've got a lot of very powerful ways of building molecular complexity in this sort of highly convergent way. So we took our biocatalytic building block, the two other, this is all color-coded to make it easy to follow, the two other building blocks, and in one step you make a molecule that is 2 steps away from the API. So that, instead of a linear synthetic route, you've got a convergent synthetic route, okay? So this offers the prospects of making this drug in a much more cost-effective manner. And in fact, we published, but we also patented this chemistry, and we've licensed this chemistry now to a third party who are scaling this as a route to manufacture of that API. Okay, but the message is we combine bio and chemocatalysis. And then the final two themes, which I've lumped together, if you're going to do this, then we need to develop biocatalysts with broad substrate scope, and importantly, that are active and stable under the conditions of the chemical process. So the phrase that people use now is, in the old days, because the biocatalysts weren't so effective, you fitted the chemical process to your rather inefficient biocatalyst. Now we can make our biocatalysts fit the chemical process, okay? And that's a very, very powerful technology. And I've got a slide that addresses that. So Enzyme Evolution, which interestingly, if you trace the history of directed evolution of enzymes, the first papers started to appear in the early to mid 1980s, and then there was an explosion of interest. And I would say this field has almost become a sort of standard method that is used now in development of biocatalysts for practical application. So in 25 years it goes from, you know, a paper in Science to something that's almost routinely used. And the reason it's routinely used is it's extremely general and it's extremely powerful. You can take an enzyme, you can make a library, you can convert it to a library of proteins, and you can screen that library of enzymes for pretty much any activity you're interested in, and then you can find the best hits and go back and do it again and again and again until out pops your optimized biocatalyst. And the main issue, I think, with this approach at the moment is how quickly you can do it. And at the moment it's okay, but it's not quick enough. So you need to, if you could do this in weeks, then the uptake of biocatalysis would be enormous, okay? Because everything's a competition, and you know, chemists can probably improve their catalysts faster than we can improve our biocatalysts. But we will solve that problem. It just takes a little bit of time. So I think this is almost the final slide. Synthetic biology is going to be a major theme in everybody's life if you work in this area, because synthetic biology, I think, brings together a lot of what I've talked about, and different people think of it in different ways, but it's going to be used, I think, almost as an umbrella term to bring in all of these technologies that are around, you know, essentially developing systems to make chemical products, including pharmaceuticals, in a much more efficient way. So it definitely includes biocatalysis. It includes directed evolution, rational redesign of proteins. It includes making sure those enzymes work on synthetically useful substrates. It really does include combining biocatalysts with chemocatalysts to do multi-step transformations, which is very much what WP4 addresses. And it also, as I said, includes combining biocatalysis with chemocatalysis. And it includes other things as well, and particularly in the context of doing a lot of this in a wholesale system to make the approach even more sustainable and efficient. So, challenges. It's my final slide. This is something we're thinking about a lot in Manchester. Can we design new and general synthetic routes to broad classes of products, the sort of things that feature in APIs? Okay, so this goes back to the point of looking at the whole synthesis and coming up with a new route. When you learn chemistry at university, in the second year of your course, you'll be taught retrosynthesis. Okay, so you'll be taught how to make molecules based on disconnections and breaking into fragments, which you can then assemble using synthetic methods. When you get to the end of your chemistry course, somebody will say there's this thing called biocatalysis, and even in Manchester, because I give this course, it's the last course that the students take before they go away and do their exams. So I think a lot of people think this has got to be brought in much, much earlier. When we teach retrosynthesis, like aldol condensations, Wittig reactions, we need to teach, well, by the way, you can do a keto reduction or you can do an aldolase reaction. Otherwise the students get completely confused because they think biocatalysis is something that's, well, we put it at the end of the course because it's not really that important, but, you know, just in case you need it when you go out there. So where are the gaps in biocatalysis? When you do this whole analysis, you find out, you know, what enzymes were lacking, and so, you know, and this is happening. The biocatalysis toolbox is expanding, but I think you need a much more strategic approach to this, rather than hope enzymes will become available. You need to do more of a sort of gap analysis and say, well, frankly, we're missing some pretty important enzymes, and they need to be developed sooner rather than later.